Okay, so I want to do a quick review of mitosis before we get into meiosis. So here we go. Remember, when we say mitosis or meiosis, what we're really talking about is an entire cell cycle, okay, where one cell, in the case of mitosis, one cell is going to become two identical daughter cells, or in the event of meiosis, which we'll talk about later, one parent cell is going to give rise to as many as 4 daughter cells, and they're all going to be different from each other. But let's get started, first of all, with the cell cycle and mitosis. Remember, in interphase, we've got 3 distinct subphases. We have G1, S, and G2. So let's talk a little bit about what's going on in each of those really quickly. In G1, as you can see here, our cell has just finished dividing. So the main focuses of G1 are going to be replicating organelles. Okay, you see all these mitochondria, and basically just recovering from the division that just occurred. You can see a pair of centrioles right here. These are going to have to be replicated so that you have another pair down here. You see you've got some what looks like smooth and rough endoplasmic reticulum. We know that this is rough ER because we can see the presence of those ribosomes, those little protein making factories right there. So we can see all these mitochondria. We see that we need to replicate some centrioles. All right, we see, not totally for sure, but this is probably a nucleolus. And look, also important, you see that the DNA has relaxed back into its unwound state. Okay, so that's G1, which is growth 1. Now, if we move on, so we drop down now to our next phase of interphase, which is the S phase. And S, remember, stands for synthesis. And when we say synthesis, we're talking about copying our DNA or DNA replication. And we've talked about this in class. We've talked about semi-conservative replication. So if I were to make a strand and open it up, okay, here is my parent strand, and here you can see it's been opened. Let's label some stuff here. Here's a 3', which means that this would have to be a 5', which means that because DNA is anti-parallel, that would be a 3', that would be a 5'. Okay, up here we've got our nitrogenous bases paired with hydrogen bonds. And remember, we always say that an A always pairs with a T, a G always pairs with a C. Okay, so now as we're opening up our bases here, okay, this semi-conservative replication means that we're going to bring in, with the help of enzymes, of course, complementary bases, and we're going to create a new strand here. Okay, and again, notice we're constructing things in an anti-parallel fashion here and here. Okay, if that's a 3', that means that this end here would be a 5', and it would be the same thing over here, but since that's a 3', we would have a 5' here, we would have a 3' there. Okay, so I'm not going to bother with filling these in because this is just a review, but you understand, you know, that if we had an A here, that there would obviously have to be a T paired with it and so on. Okay, so in the synthesis step of DNA replication, or in the synthesis step of interphase, okay, the S phase of interphase, we're replicating the DNA. All right, G2, this is our final checkoff before we enter mitosis, before we actually start dividing the nucleus. This is our final check, because once we proceed past G2, we can't stop, we can't go back. So we're going to make sure we've got all of our organelles replicated, all of our cytoplasm's taken care of, all of our organelles are good, everything else is ready to go. And that then takes us to prophase. So we look at prophase, and what do we need to point out? Well, a couple of things. First of all, our nucleolus has broken down. Remember, the nucleolus is a dark stained spot. It would be kind of, right here in the nucleus, just maybe like that, but the nucleolus has broken down, so it's gone. Okay, there, it's gone. Chromatin is condensing down into visible chromosomes. Remember, you can't see DNA, and it's really even hard to see chromatin, but when we take that DNA and we wrap it around protein, DNA condenses to chromatin, DNA to chromatin to chromosomes. Okay, sorry about that. So, there we go. DNA condenses to chromatin, which condenses to chromosomes. Let's fix that arrow real quick. There we go. Okay. So the chromatin condenses into chromosomes. You can see here and here. This is a centriole. This is a centriole. This is a centriole. This is a centriole. But the entire complex I circled is a centrosome. They start migrating to opposite ends. Okay, and you see that the mitotic spindle is starting to form as well. You also see what looks like the first beginnings of perforation there in the nuclear membrane. Okay, so now we jump down to prometaphase, and you see that now the nuclear membrane has totally broken down, which makes sense. It's got to. If we're going to move chromosomes to opposite ends of the cell, we've got to break down the nuclear membrane. You can see that the chromosomes have replicated, and remember that happened back in the S phase. So you need to recognize that these are replicated chromosomes. Replicated chromosomes. And the little black dots there are kinetochores. And those are going to be little handles for the motor proteins that move up and down the mitotic spindle to grab onto and then start moving those chromosomes to opposite ends of the cell. Okay, so we're in prometaphase now. Nuclear envelope's gone. The mitotic spindle is moving out towards the center, and I think we've covered that well enough, so let's jump on. Metaphase. Okay, remember, when you hear metaphase, you need to think middle, because that's where our chromosomes are at. They're lined up along this imaginary, that's terrible, they're lined up along this imaginary equator. Okay, here we go. They're lined up on this imaginary equator, and we're about to start doing some real work here. We're about to start dividing these things out. Okay, so the chromosomes are here. They're on this imaginary metaphase plate. The mitotic spindle, and remember this too, here's the mitotic spindle. There's a motor protein that walks this thing, okay, that walks this chromosome down the mitotic spindle. This stuff doesn't just happen. Okay, here would be 1/2, so there and there, and let's put a kinetochore there, and that motor protein walks that thing back. Okay, it uses ATP for energy, but these things don't just just randomly move on their own. It's walked back. Okay. That happens, though, in anaphase. So we jump up. And here we go. Okay, anaphase. You can see now And this is happening in two ways. One, like I said, the motor proteins are pulling these guys back. They're walking them back, rather. So if I had a mitotic spindle, and I'll try to match the colors up. If I had a mitotic spindle, a little bit right like that, okay, a little bit of cytoskeleton, I would have a motor protein that's walking that thing back. And let's make my little motor protein in pink. So there it is, and let's make my chromosome here in orange to kind of match that one there. So there it is, and there's that kinetochore. And again, it's drawing it back this way. Now, also what's happening here, though, is this entire apparatus, these spindles are being shortened. They're being shortened down here at this end. So the motor protein's walking it back, but the spindles are also being shortened at the other end. Okay, so that's anaphase. And you remember, the easy way, if we remember that metaphase is middle, we remember anaphase equals away. All right, so after anaphase, telophase. And telophase is pretty easy. Basically, everything that we did in prophase and prometaphase, we reverse here. So, prophase and prometaphase, we broke down nuclear envelope. You can see we're replacing it already, and we're doing that in kind of a green color here and here. The chromosomes are relaxing. You can see here we had some red ones, here we had some orange ones, and you can see they're already starting to relax and unwind back into chromatin. The nucleoli are going to reappear. You can see a red one there, you can see an orange one there, and the cell is still elongating. Now, what's actually happening here, and they don't do a good job really of showing this, but again, stuff does not just happen. The way this is elongating is you have some of these microfibers and microtubules, and they're actually lining up and connecting to each other and pushing each other away. Okay, they're pushing each other away. And that's what's making the cell elongate. This stuff doesn't just happen. It requires energy, it requires ATP. So that's how that's happening there. All right, so we're through with telophase. And that brings us to cytokinesis, which is the division of everything else. We've created two brand new nuclei. Okay, now we again are going to use microtubules, microfilaments, and we're basically going to form kind of a belt around a portion of the cell, and we're just going to start tightening that belt down. And that creates this indention here, and we call that a cleavage furrow. And eventually that's going to get smaller and smaller and smaller until this pinches off into two identical daughter cells. And that is very, very important. The whole purpose of mitosis is to give two identical daughter cells. The DNA should be exactly the same because we did this with semi-conservative replication. So that's a quick review of the cell cycle, and I hope that's helpful.